Okay, the next presentation is by Oliver Hawlitschek. He's a scientific manager of the Molecular Genetics Lab at the Centrum for Natuurkunde in Hamburg. And his key interests are evolutionary biology, biodiversity research, and species conservation. Although he did study other organisms, everybody can make mistakes, of course, like water beetles and reptiles, he's working already for quite a few years on Orthoptera, and today he has a question for us about phylogenomics of Acrididae. Oliver, please.
Yeah, thanks for the introduction. Yeah, I always get hate. I get hate from the herpetologists because now I work with insects, and I get hate from the entomologists because I worked with herbs and other things. So yes, maybe I'll just add a little to the introduction. Forward. Yes. You already know me. I'd like to quickly introduce Edgardo and Ricardo, who worked with me in this project. Edgardo is a bioinformatician at the Technical University of Munich, and Ricardo is an evolutionary biologist at the University of Munich. Yeah, and the background of this project was my earlier work on the barcoding of orthopterans in what they call Western Central Europe. So 3 countries, Austria, Germany, and Switzerland. The goal was just to establish a barcode library in the hope of this allowing us to identify species based on DNA barcodes, which worked in some extent, but one of the main findings, was that we could not identify all species by DNA barcoding. In fact, there were quite a lot that we could not identify. Initially, the problems are due to the DNA barcode per se, because we use the cytochrome C oxidase 1 marker, which is a mitochondrial gene, and the mitochondrial genome may not be representative of the whole genome. It may have a different evolutionary trajectory. And also, it is just a very tiny part of the whole genome. The most obvious case in which this did not work out is what I call here the Cordyceps bicuculus complex in the context of our barcoding study, which is this assembly of very notorious species, Cordyceps bicuculus, Bruneus mollis, and interesting for us, we found that we could not even distinguish them genetically from Cordyceps apricarius and even Gomphosorubus rufus, which is a whole different genus according to current taxonomy. As we all know, we can distinguish these species using songs, for example, and morphology, but with the DNA barcoding, we found this was not possible. There may be some reasons for that. We try to speculate at least what might cause this, and one reason is that these were assumed to be very young species, evolutionary, very young species. So they had probably developed into different species only, possibly during the Pleistocene, during the glacial cycles. In some cases, as we know, hybridization is still possible. So this may cause the integration of mitochondrial genomes from one species into the other. We also know that orthopterans, especially acridids, have very large genomes and with a lot of pseudogenes. Pseudogenes are copies of genes that are non-functional but can be sequenced instead of the ortholog of the actual gene, and this may cause trouble in the barcoding. And finally, Wolbachia infections may play a role that cause various kinds of genetic chaos, I would say, that makes it more difficult for us to study these groups. Yeah, based on this, we started asking some questions, such as if we just use genomic data, more data especially, will this be more informative? If we do this, will we find different results? So more informatives, or will we also find that maybe the results we had so far based on mitochondrial data are misleading compared to the nuclear genomic data? If we find this, what will be the taxonomic consequences? So what we did in this small project is that we used transcriptome sequencing, which I will explain, with the example mostly of the Cortipus bigutulus and related species, but also including some other acridid species, just to be able to see if the same holds true for other species too. What do we do for transcriptome sequencing? Well, obviously we start with a sample of, in this case, a grasshopper. Just a quick repetition, the transcriptome. So we have a genome, obviously, which is all the genetic or genomic information that an individual has, which consists of genes and much other material. The genes, the actual genes that code for proteins, only make up a small proportion of the whole genome. If we want to specifically target genes, the transcriptome makes sense because the genes are transcribed into messenger RNA, and this is then translated into proteins. And by extracting the messenger RNA, we get a set of the genes that are currently active and transcribed. This can be sequenced, and this will hopefully result in data useful for phylogenetic reconstruction based on hundreds or thousands of genes. Not only one gene, as in barcoding, or not only 16 genes, as in a whole mitochondrial genome, but hundreds of thousands. Ours is not the first study. This has been done by several people, for example, in the study of Burden et al. 2015 by Frida Meyer, already on Cortipus bigutulus. And a more recent one is the study, the phylogeny of Orthoptera as a whole, by Song et al. 2020, and there's more. But so far, our group has not been included. So we did the transcriptome sequencing. Nevertheless, we started with the mitochondrial genomes, which were also included in the transcriptome because we wanted to see if we already found some information there. We also used this for a molecular clock study based on the complete mitochondrial genomes. And especially here, I think you can hopefully read this. Well, the first thing we see is that this is largely congruent with DNA barcoding in that we still cannot distinguish these species. So you see that there is bigutulus, there's mollis, bigutulus again, mollis down here, rufus in the middle, then Bruneus down here. So this is basically a big mess in which all these species are at varying positions. Nevertheless, we used this mitochondrial phylogeny to use a molecular clock and confirmed what we already knew before, but also based on our data, that this Cortipus bigutulus complex, so here there's a mean age of 1.38 million years with some standard deviation. So anyway, it's certainly younger than 2 million years old, which partly explains that at least the mitochondrial data has just not been evolving separately for long enough to show clear distinctions. But what we were more interested in was the phylogeny based on those 540 nuclear genes that we put together here. This is just the overview of the nuclear genes and the mitogenome, and the straight lines highlight species that are in the same place, and these crossing lines highlight species that are not in the same place. So the first thing we found is that these were incongruent. We found big difference between this whole phylogeny of the nuclear, of the many genes, and the mitochondrial phylogeny. Yeah, I think this should be visible here. So one thing we find here in the coalescent tree is, or the nuclear tree, is that this may have to be confirmed by adding more specimens, but at least here with the few we have, we find that all the species of the Cortipus bigutulus complex become monophyletic. So this is Cortipus mollis forming one clade, this is Cortipus bigutulus forming one clade, Eisentraute here. Okay, we only have one of Tibialis, we have two of Bruneus forming a clade, and so on. Rufus, Gomphosorhynchus rufus, is still here, is now the sister group of this complex. Yeah, and then Apricarius is finally outside. And this is different to what you found in mitogenome. So this may show us that the mitogenome may just have evolved differently and may not be a very good representation of the whole evolutionary history of these species. So that shows us that this genomic data is more informative than only the barcodes and the mitogenomes. However, we have a very limited sampling here. We just included a few species that we could easily get hold of and made a preliminary phylogeny. This does not yet allow us to draw any good taxonomic conclusions. Some are pointed to, but we don't have a very comprehensive data set yet. So what we're currently doing is we're increasing our sampling across Gomphosorhynchi, mostly including the type species, Gomphosorhynchi. Not only, in fact, we want to include as many species as possible, Cortipus and other genera, and also other groups of acridids. We want to use transcriptomes, for which we need fresh data, but we also want to use historical samples because it's not always easy to get hold of fresh samples for all species. And we will use targeted capture sequencing, which is a bit more complicated than transcriptomes, but can be applied to historical samples too, with the goal of, in the end, having a good taxonomic revision of this group sometime in the future. Also, what I would like to do personally is find a single marker of any nuclear gene that can work for barcoding, that will allow us to distinguish these species, because it should be there somehow, but we haven't found it yet. So far, I've just checked those 540 genes, but so far, only combinations of many genes allow the distinction of these species, not a single gene yet. Well, but I keep searching. Yeah, I have a lot of people to thank for, some of whom are here, and the funding bodies, and thank you for your attention.
Thanks very much, Oliver. Who has got a question?
Thank you, Dragan again. I have two questions, one just for personal use. How did you calibrate it for the molecular clock of the mitochondrials?
Yeah, it's not a very good calibration. We just extracted the calibration points from the phylogenies Pahojun Song, which were in turn calibrated by fossils, but fossils which are pretty far out groups of what we have here. We don't have any good internal calibration points. So yeah, this is as close we can get with the calibration. The other option would be rates of mitochondrial evolution.
Thank you. And another one, you said you believe you will find the gene that could separate. I doubt it a little bit. Maybe for certain groups, yes, but in general, maybe some SPNs from transcriptomes. And how do you see the future of this transcriptomics to be used in the barcoding of the organisms? Thank you.
Well, I think with the further development of technology, I think we'll be able to, at some point, not necessarily replace barcoding. We will have tools that allow us to use more genomic data for species identification. The problem is first, this is already becoming more and more affordable, cheaper and cheaper, but the analysis is not yet straightforward. So this will only be possible at the time at which a very easy analysis is possible. You just click a button and then you identify a species using the genomic data, which works for barcodes now, more or less. You just paste the sequence into the database and then it'll identify your species. But this is not yet possible with genomic data. As of now, it still takes weeks or months of analysis. And only once this is possible, we'll be able to use this instead of barcoding. And the second thing is that the crucial point is the reference library. So there's a huge reference library of millions of specimens of many, many species for DNA barcodes, but not yet for genomic data. So this would also have to be established anew, and this will take, yeah, this will, if it will ever exist, it'll be years, decades in the future, I think. But the development is there.
Okay, I think we have to go to the next. I'm sorry, we have to go to the next presentation. Thank